Hello, everyone. Welcome to today's live broadcast, Advances in Twin Screw Granulation Technologies. I'm Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be moderating this event. We're pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Thermo Fisher Scientific. I'd like to share a statement from our sponsor. Thermo Fisher Scientific is the world leader in serving science, with annual revenues exceeding $30 billion. The company's mission is to enable their customers to make the world healthier, cleaner, and safer. Whether their customers are accelerating life sciences research, solving complex analytical challenges, improving patient diagnostics and therapies, or increasing productivity in their laboratories, Thermo Fisher Scientific is here to support them. Their global team of more than 80,000 colleagues delivers an unrivaled combination of innovative technologies, purchasing convenience, and pharmaceutical services through their industry-leading brands, including Thermo Scientific, Applied Biosystems, Invitrogen, Fisher Scientific, Unity Lab Services, and Patheon. For more information, please visit www.thermofisher.com. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, click on the question mark Help widget in the dock at the bottom of your presentation window. Now I'd like to introduce today's speaker. Professor Dennis Doromis obtained a bachelor's degree in chemistry and a doctorate from the Department of Pharmacy at Patras University in Greece. He worked as a postdoctoral fellow at the Friedrich Schiller University of Vienna Department of Pharmacy for the development of parenteral formulations of anti-epileptic drugs. He also worked as a team leader in the development of nanoliposomal formulations in the treatment of rheumatoid arthritis by applying photodynamic therapy. Following this, he worked for Focus Pharmaceutical as a senior scientist at the Product Development Department. Thank you for joining us today, Dennis. I'm going to turn the presentation over to you.
Hello, everyone. I'm Professor Dennis Doromis, and my presentation today is about advances in twin screw granulation. I will start with the principles of twin screw granulation and related mechanisms, and then I will continue by presenting the state-of-the-art systems on continuous granulation. Then I will present a few cases on twin screw granulation related to modeling of the process, and finally I'll present some cases where quality by design approaches are implemented, usually coupled with process analytical tools. Before I start, I would like to tell a few things about our group. We are based in Medway campus of the University of Greenwich. This is southeast UK in Kent, and our group focuses in continuous manufacturing and PAT tools by using a range of different formulation technologies, mainly hot melt extrusion and twin screw granulation. We also have a keen interest on 3D printing of medical devices by using a range of analytical 3D technologies. And finally, we focus on nanotechnology by developing drug delivery systems for passive and active targeting of cancer. Granulation is well known in the pharmaceutical industry for more than 50 years, and it's a process that usually involves the enlargement of fine powders in which small particles are agglomerated, compacted, or brought together into larger, relatively permanent sizes. The granulation offers several advantages and can be used to improve the flowability of powder blends, improve the homogeneity between the drug substance and excipients in the formulation, controls the particle size distribution to prevent the segregation of the material during downstream processing. It has been used also to enhance the tableting of powder formulations and also regulate the dissolution of drug substance. Over the last 13 to 14 years, there was a strong interest from both academia and pharmaceutical industry to exploit twin screw granulation process and optimize it for the commercialization of marketed products. However, twin screw granulation was invented by Gamble and Erdely in 1986, and they developed paracetamol granules by using the wet granulation process. A year later, Lidberg, and in 1998, Kleine-Budde also employed twin screw extruders for the formation of effervescent granules. There are basically three types of twin screw granulation. The first one is wet granulation, where by adding granulated liquid, this can be aqueous or organic, we use to form powder blends and produce granules. There's a melt granulation process where a molten binder is used for particle enlargement of powder excipients. And lastly, dry granulation, which is very similar to melt granulation, but only the kneading zones are heated. An interesting part for twin screw granulation is that it uses exactly the same equipment which is typically used for whole milk extrusion or from food industry or plastic industry. And this is the twin screw extruder. As you can see on the top right image, this is an 11 millimeter twin screw extruder, which comprises of two main screws, and each screw is comprising from different screw elements which are assembled on high torque spring on these high torque spring shafts. The shafts are placed in barrels where we can control accurately the temperature during the process, and basically this is the main principle where we can apply twin screw granulation. Twin screw granulation processing is controlled by a variety of processing parameters, and some of them are listed below. So the very first one is the barrel temperature. We know that the heat transfer between the barrel and the formulation affects the product quality, and usually we use temperatures that vary between the melting point, the glass transition of the polymer, but usually below the melting point of the API. There's a strong effect of thermoplastic binders due to low melt viscosity, and it has been found that by applying higher temperatures, we have a faster agglomeration, stronger granules, which have large size and are less friable. Also, the screw design and screw configuration are very important because they have a significant effect on the size, strength, structure, and temperature of the granules. The increased number of mixing zones results in oversized granules with less fines, and usually large offset angles of kneading elements provide again larger and denser powders with less friable powders. This is mainly because of the high shear and stress that is applied during the granulation process. The angle of the kneading elements has an effect when filling degree of the barrel is greater than 70%, and it has been found that by using 90-degree elements, we have the creation of lumps or the blocking of the extruder, and hence this should be avoided. On the right image, we can see a representation of different screw elements with some conveying elements and kneading blocks with different offset angles. These elements can be used in combination during granulation process, and we will see later the quality of the products can be controlled. The increase of the kneading elements increases the barrel temperature due to the created friction, and also has been found that kneading elements produce elongated shapes compared to spherical shaped granules which obtain when we only use conveying screws. The use of conveying elements results in highly porous and friable granules. This is something you can easily see on the right top graphs, where we have a representation of the porosity when using different elements. So, for example, we have a high porosity of the granule when we use conveying elements with a pitch size of 15 millimeters, followed by 20 millimeters, 30, 40, and so on. And eventually, the less porous granules are formed when we use kneading elements. The same trend we can see in the middle graph, where it counts for friability. Kneading elements provide free-flowing powders, while conveying elements easy-flowing granules depending on the pitch size. Lastly, the feeding rate and the screw speed are very important, and it has been found that high feeding rates are related to increased compressive forces, which correspond to larger, denser granules. High feed rates, when with no kneading zones, produce friable granules, as we mentioned before, but also it has been observed that there's a non-uniformity on the liquid distribution. Also, there was no direct impact on the screw speed on the size granules. However, the combination of the feed rate and the screw speed can be adjusted to the degree of fill and thus the granule size. This can be seen on the bottom graph on the right, where we have the particle size update from two different degree of fills of the extruder. So we can see that the higher the degree of fill, when it's around 70%, we obtain larger particle size of the granules. As we mentioned, the processing parameters are very important on the quality of the produced granules. However, there's an important effect of the materials that we use to form the granules. The binders, which are usually classified as hydrophilic and hydrophobic, they have a strong effect on the produced granules. And for immediate release dosage forms, we use hydrophilic binders, which are usually of two types: the low melting point binders with low viscosity, such as polyethylene glycol, or thermoplastic binders with high viscosity. For melt granulation, we can use thermoplastic binders with high melting points or higher glass transitions, and usually low molecular weight results in weak and friable granules. But it has been found that it facilitates granule growth and spreading. High molecular weight of binders results in higher viscosity and, of course, stronger granules. Overall, the effect of the binder depends on the formulation composition, as we see later on in some of the examples. On the right, we can see a table which shows the most frequently used polymers and materials as binders, and we can in the last two columns, we can identify the glass transition temperatures or the melting temperatures of these materials. The binder particle size affects also the granule size. So, for example, when we use larger particles of a binder grade, we produce larger granules. Thermoplastic binders with large size can result in incomplete process. What is very important and has been studied from other researchers is the drug binder miscibility, because when the drug and the binder are miscible, we can process the materials using lower temperatures due to the pasteurization effect. However, it has been proved that the high miscibility between the drug and the binder creates a discontinuous phase with pure distribution compared to immiscible systems. The table below shows the formulation and process variables that affect the physical attributes of the granules. So it's a summary of all these effects that have been studied over the years. The next part of the presentation is related to the mechanism of the granule formation. Usually the granules are formed through a combination of capillary and viscous forces, facilitating particle binding in the wet stage. When the wet material is distributed, compacted and elongated in the kneading elements, and eventually the transformation of the particle morphology from a small to a large, irregular and porous granule takes place. Other sub-processes such as aggregation and breakage can also take place during granulation. On this schematic graph on the right side, we can see how they are formed, and we can take an idea of the granule mechanism. We start the binder liquid, where we have the precursor molecules. We see the creation of nuclei in the first kneading zone, and followed by the growth of the granules, aggregation, layering, or breakage, and eventually the consolidation, which are usually take place after the kneading zone. The wet granulation mechanism was further studied by a group of researchers, where they split a granulator in 5 different sections. The powder addition was done in the beginning, where they use a large pitch conveying element screw, and later on in compartment C1 they use small pitch conveying elements to avoid back feeding of the material. They observe that the nucleation occurs in C1 compartment where we have bigger size agglomerates, and as we move to section C, which is the kneading, the first kneading zone, we had consolidation and breakage. In this case, they observe a reduced particle size, and by moving in compartment C3, we observe coalescence and breakage where the increase or decrease of the particle size took place. The actual decrease or increase of the particle size in compartment C3 depends on the processing settings and the actual formulation. Also, the coalescence and the consolidation took place in compartment 4, where a further decrease in the granule size and the final breakage took place. What was observed in this study was that in compartment C3 and C4, there was increase of the granule strength, indicating more consolidation and coalescence. And eventually, the reduced particle size of the produced granule was observed in compartment C5, which also comprised of small pitch conveying elements. Melt granulation mechanism has been also studied, and it was found that primary relates to two different mechanisms based on the viscosity and the size of the binder. The first mechanism is well known as the immersion and layering mechanism, and during this process we have the spreading of the liquid binder over the surface of primary particles with the nuclei formation occurring by coalescence between those welded solids. The favored mechanism takes place when the binder particles and droplets are small related to the surrounding powder matrix or when the viscosity of the binder is low. The second mechanism is known as distribution and coalescence, and the nuclei are formed by primary particles being captured onto the surface of the droplet of the binder and they later start growing around the granule as the binder is drawn to the outer surface. The mechanism appears when the binder particles are large and usually larger than the surrounding powder matrix, and the binder exhibits high viscosity. The increased binder particle size results in larger granule, and eventually it was concluded that the dominance of the two different mechanisms depends on the ratio between the size of the solid particles versus the binder droplets, the binder viscosity, and the intensity of the mechanical agitation. Over the last 20 years, there was a lot of interest in material manufacturing, mainly from pharmaceutical industry, and this has been supported strongly by the regulatory agencies such as FDA, EMEA, PDMA, or MHRA. And basically, continuous manufacturing is a method of manufacturing products and processing materials without interruptions and with constant material feed and removal. The continuous manufacturing offers several advantages over traditional approaches, and some of them are listed on this slide. So, for example, it is used to reduce risk of manufacturing failure and prevent drug shortages. It provides flexibility by using the same system to develop a manufacturing process. It also provides increased supply speed and react to market demands. It offers agility and reduced scale-up efforts. One of the key advantages is the real-time quality assurance, whereby using PAT tools, we can have inline or line monitoring of the process. And finally, there is a reduction of the capital and operational costs, and usually these continuous manufacturing systems have a small footprint. Continuous manufacturing was one of the attractive technologies that have been exploited by pharmaceutical industry, and twin screw granulation was one of the first that was used to optimize and develop continuous lines. One of the continuous systems was developed by GEA, and it's called Ecosigma, which is a continuous oral solid dose granulation system combined with drying and tablet compression. It integrates a multiple unit operation, and it can be used both for R&D and clinical batch manufacturing. The Ecosigma comprises of different steps. We start with the dosing and mixing of raw materials, wet or melt granulation, which is the second step. And during that, we use a twin screw granulation unit. and subsequently the granules are continuously transferred by vacuum or gravity to a fluidized bed dryer. The drying takes place either by cooling of melt granulation or the removal of the water when we apply wet granulation, and the system is monitored by a moisture probe. The next step is the granule milling by using an inline mini blender, and eventually we have the tablet coating, and usually this is the end of the process. One of the major advantages of Ecosigma is that it provides a quality control system where a lot of these stages are covered with inline PAT sensors. In this slide, we can see a photo of a Ecosigma platform line, which can be used for the production of granules and eventually the end product in the form of tablets. And also at the bottom right schematic diagram, we see the full operation of the granulation system. Another continuous manufacturing line was developed by GLAD, and it's called MODCOS, which is a fully automated continuous wet granulation platform and operates both in a batch or continuous mode. It converts a batch fluid bed system to a continuous fluid bed drying system, and the technology provides narrow testing time during the drying step because of the use of a sophisticated discharge system. The MODCOS system is fully integrated with PAT tools and comprises of NIR probe, which can be used to investigate and record the granule uniformity, a moisture probe during the fluidized bed drying, and a particle size probe to record the particle size of the obtained granules. In this slide, we can see a schematic diagram and also a full, a complete line of the MODCOS platform that has been used for the optimization of granule formulation and eventually the end product in the form of tablets. As we mentioned, the continuous systems offer some advantages. Some of them are already mentioned in the previous slides, such as flexible batch size with no need for scale-up. These platforms are compact, offer a modular construction with very short installation time. They provide consistent granule quality with improved compressibility characteristics. What is very important is that similar investments have been made by other companies such as Ferti and Synergon. And interestingly, Synergon focuses on the production of many batches using a series of small parallel fluid bed granulators and dryers. So this is a completely different approach, and Synergon does not focus anymore on the classical continuous manufacturing process. On this table, you can see a list of products from different pharmaceutical companies that have been marketed by using twin screw granulation systems. As we can see, most of these products were marketed in 2015 and onwards. The next part of the presentation is about computational modeling. I will present some cases where researchers try to investigate the twin screw granulation process and combine that with computational modeling in order to evaluate the process, provide better understanding, and eventually predict some of the parameters. In the first study, there was implementation of a conceptual model to better understand and simulate the residence time of the process, where the experimental data were used to estimate the parameters of the model and to analyze and predict the effects of changes in the number of kneading discs and the Stanger angle, also the screw speed and the powder feed rate. So in this case, the authors used three different models. The first one was the tanks in series model without plug flow and volume fraction. In the second model, the tanks in series with the plug flow volume fraction, and the last one, the tanks in series with the plug flow volume fraction and dead zones. The residence time was defined by the two equations we see on the right side of the slide. And this is the schematic representation of the tanks in series and the approach that was used for each of these three different models. The system with the plug flow volume fraction, the dead zones, showed a very good fit between the experimental and estimated resident time data, and it was concluded that was the best model to describe the process. As you can see, the R square varied between 0.93 and 0.99, and the volume fraction of the model increased with the number of kneading elements and the staggering angle, which both of them were found to be critical. The axial mixing was estimated indirectly by the number N of the stirred tank reactors, and it was found that when axial mixing takes place, we have broadening of the residence time distribution. The data analysis indicate that powder feed rate and screw speed together dictate the axial mixings, and it was concluded that a balance between the throughput force and the conveying rate is needed in order to obtain a good axial mixing during the twin screw granulation. High feed rates and increase in stagger angles reduce the axial mixing. The values were equal to 4, even when screw speed was increased. Usually, when the axial mixing is increased, the number of tanks is reduced and is equal to 2. The number of dead zones was affected by the screw speed and the number of kneading discs, and increased kneading discs reduced the bulk mixing, while at the same time increased the fill ratio, which is the feed rate, and eventually reduced the stagnant region and held the dead zones. On this graphical, on this graph, we can identify and see the effect of different process parameters: the screw speed, the stagger angle, and the number of kneading elements, and their effect on the dead zones. As you can see, the dead zones are reduced when we increase the number of kneading elements, that was observed during the study of the system. On this graph, we can see the representation of the residence time for a wide range of experiments carried out during the study. The dashed lines represent the predicted model, while the solid lines the measured residence time. We can see that, as mentioned before, there's almost a perfect fit between the experimental and the predicted values of the residence time. In the second study, a multi-component population balance model was developed, and that was based on the processes such as aggregation, breakage, liquid addition, consolidation, and powder flow. This two-level design was used to track the particle size, liquid distribution, and granule porosity. The twin screw granulation process is divided into 3 axial zones using a compartment approach. This is obvious on the schematic diagram below, where we have the powder feed and the pre-mixing zone. Then we have the liquid addition, where we add the liquid binder, and the last zone is the wet massing zone. And the different elements of the process, such as the particle size, the liquid content, and the gas content, are defined by the equation we see on the slide. And similarly, other elements during the process, such as the flow, the liquid rate addition, the consolidation, and the breakage, both for liquid and gas, were defined by similar equations. For the interpretation of the fitted and the predicted data, the authors used different kernels, and they tried to match the particle size distribution of the obtained granules. As you can see on the top 2 graphs, the 2 different kernels gave a perfect fit for the D25 and D50 of the obtained granules, but both of them failed to predict the D75 of these granules. This is also obvious in the middle graphs, where we have the R-square values for each of these granules. However, the predicted porosity was really good, and they matched the experimental data. The R-square was 0.984, and we can see at the bottom graph the fit was quite perfect. The model was also used to study the simulated dynamic behavior, and on the top image we can see that the process was studied over time, and the D50 of the obtained granules was recorded. What we can see here is that the granulated liquid started adding at 5 seconds, and the steady state for each process was obtained at 25 seconds. This is very interesting because it was found that the steady state reaches at the same time for different liquid-solid reactions, and it was independent. The bottom image shows a graph where the process was studied over time and the particle size of the granule was recorded. In this case, it was found that at 4 centimeters from the start of the liquid addition zone, the median diameter increases slightly. We can see that one, and it doubles at about 10 centimeters after the liquid addition. Eventually, after 20 centimeters of the liquid addition, we see the particle size enlargement of the produced granules, and this is where we reach the steady state of the system. By design approaches have been implemented by pharmaceutical industry and are also strongly supported by regulatory authorities, and usually those are coupled with PAT tools. In this part of the presentation, I will present some case studies where quality by design approaches were implemented, and the process was studied in order to have a better understanding of granulation, but also to optimize the process. The quality by design approach was used by introducing the Ecosigma powder-to-tablet wet granulation line, as you can see on the top right image, and the whole process was coupled with PAT tools, where NIR Raman probes were used at different stages of the line, and also spatial velocimetry for monitoring the particle size of the obtained granules. The aim of the project was to evaluate the variation in critical raw material properties on the critical quality attributes of the granules and the compressed tablets. For the purpose of the study, theophylline, lactose, and PVP were used as model formulation at different ratios, and during the study, a two-level full factorial design was introduced. As you can see at the bottom image, the screwed configuration and design comprised of two kneading zones. On the left table, we can see a range of product variables, and those are categorized according to the equipment variables, the starting powder properties, the liquid characteristics, and the process variables. These are, as we can see, there's a range of different properties that we can take into account when we optimize the granulation process, and the number is almost unlimited. However, for the cases of this study, the authors use only a limited number of independent variables. So, for example, they investigate the screw speed, the powder feed rate, and the water content, also the barrel temperature across the granulation process. So the screw speed varied from 600 to 950 RPM. The powder feed rate varied from 10 to 25 kilograms per hour, and the water content also was defined at different levels. A typical DOE experiment resulted in large and oversized granules, and this is obvious on the top left graph, where we can see the obtained granule particle size distribution of one of the experiments, and we see that there is a small amount of fines, but we have a wide range of larger particles of the granules. The powder feed rate and the barrel temperature have a significant effect, and which is usually positive, on the D50 of the obtained particle size of the granules. So usually high powder feed rates and higher temperatures lead to increased particle size, and vice versa. The authors observed that high temperature reduced solubilization of lactose and the materials, and thus increased the bridge formation. On the middle graph, we can see the plot, the effect plot, where the different processing parameters have a significant different effect. And for example, we see the powder feed, the barrel temperature, that have an important effect on the obtained particle size, and eventually the liquid addition has a less effect. As you mentioned before, the whole process was coupled with Raman and NIR approach in order to monitor the granulation process. The Raman spectroscopy was initially used to discriminate between the anhydrous theophylline and the hydrated theophylline because of the difference of the absorption peaks between the two different forms. By recording the different runs and also the process, the experimental process, including all the DOE experiments, the authors designed PCA plots for screw speed, barrel temperature, liquid concentration, and powder feed rate. And the theophylline solid state is expressed in the PC2, where positive parts are assigned to anhydrous theophylline. Similarly, the NIR analysis of the granules that were produced with a higher liquid amount and lower temperature are spread in the negative part of the PC2 axis, while the granules that produced with low liquid and high temperature are in the positive part of the PC2. There was no clustering observed for screw speed of this processing parameter, and that was one of the interesting findings of the study. This is a summarized table that accounts for all the experimental runs of the DOE design, and it shows that the significance of each of the studied parameters for each experimental run. So, for example, we see that for the first experiment, only the water content had a significant effect, while for number 8 run, all three different parameters had a significant effect. Also, the last two columns represent the Raman NIR analysis, which shows the runs where the anhydrous theophylline was detected by using both technologies. The process was also used for the further evaluation of different theophylline grades, and in this particular study, 7 different grades with different particle size were used to obtain granules or tablets, and the effect of the theophylline particle size was studied. Granules produced with fine theophylline powders had a higher bulk density and a lower tab density, but as we can see on the top right graph, we have granules that produce by using large particle sizes of theophylline, and usually those are they present high tab and bulk density, but the obtained granules presented lower tab and bulk density. For theophylline grades where small particles were used, the obtained granules presented higher bulk and tab density compared to the physical mixture. Eventually, no tab volume reduction could be achieved by granulation of powders with high tab densities, and the granules produced with wet twin screw granulation tend to be more needle shaped. This is obvious on the bottom image, which shows a summary of photos taken after the granulation process. On the right part, we see needle-shaped granules obtained from the bigger fraction of the collected granules, but on the left part, we see more spherical morphology of the lower fraction size of the obtained granules. The Raman analysis showed some very interesting results, and it was detected that more theophylline is distributed in larger granules, while more lactose can be found in smaller granule sizes. The tablets made by granules with micronized theophylline showed lower porosity and higher tensile strength, but also tablets made by large granules of theophylline presented higher friability and vice versa. The tablets with higher porosity showed fast degradation times, and overall the theophylline bulk powders with high bulk and tab density showed very good flowability and eventually resulted in high torque with increased barrel temperature and also produced larger granules. The friability results and the tensile strength of the tablets are depicted on the right. Also, the PCA plot was designed, And what we can observe, we are not going in detail and discussing in depth this Pca Plot, but we can see that there's a clustering of different powders, and there are three different areas of where the clustering takes place. The powders of theophylline, including granules and also the tablet properties that cluster in a specific area, they denote that there is a similarity on these properties of the materials. When there's a distance between the clustering, that means that we have dissimilarities of the different powders that were studied during the process. I'll present another case study where a quality by design approach was introduced, and in this case, crystalline granulation by using water-insoluble drugs took place. Ibuprofen was used as a model API, but the interesting part of this study was that the author used inorganic excipients, combined them with the polymeric materials and polyethylene glycol as a binder. This was a wet granulation process, and what was used is a granulated liquid. As we can see at the bottom table, there were three different process independent variables that were defined by during this study. The first one was the inorganic polymer ratio, the binder amount, and the liquid-solid ratio, while the process dependent variables that were studied at the end of each experimental run was the release of ibuprofen at 120 minutes, the D50 of the obtained granules, and also the specific surface area of the granules. On the right image, we can see a photo of the granulator of the granulation process. It was a 16 millimeter extruder coupled with a peristaltic pump, and the addition of the liquid was taking place right after the feeding zone of the extruder. On the right graph, we see the particle size of the granules obtained by laser diffraction, and we can see that the particle size distribution depends on the processing settings and parameters. And for some of these formulations, the particle size appears to be monomodal with a good particle size distribution. In other runs and other formulations, we observe that the particle size is bimodal, or in the bottom graph, we can see that we have the appearance of fines at different sizes. During the study, ibuprofen remained partially crystalline, and it was found also to be in an amorphous state. And this was attributed to the effect of the binder and also the mucilin because of the API that is absorbed on the surface of the inorganic excipient. The degree of amorphicity varied depending on the twin screw granulation processing settings, and a further analysis of the granules by using DVS showed a significant water uptake for the granules with high ibuprofen crystallinity or PEG amounts. It was very interesting, but eventually the process was reversible, and there was a complete water loss at the end of the process. The granules with higher amorphous content showed less water uptake. Eventually, the granules obtained during the study provide stable formulations under accelerated stability conditions at 4 degrees Celsius and 75% humidity. The DOE regression analysis shows some interesting results. So, for example, the ibuprofen release was found to be significantly affected by the ratio of the inorganic material and the polymer, and also it was found two-way interaction between the polyethylene glycol and the liquid-solid ratio. The loss of brine was the other dependent variable that was studied. It was found to be affected by the binder and the liquid-solid ratio, which had a significant effect. The particle size distribution was also affected by the ratio of the inorganic and the polymer excipients, the binder amount, and which was found to have a significant effect. The same experimentation was repeated by the same group, where this time ethanol was used as granulated liquid, and the regression of the DOE showed completely different results and effects of the independent variables. So we can see that the granulated liquid has also an important effect on the formation of the granules. This is another study where quality by design approach was used in order to study in depth the critical processing parameters and the critical quality attributes using twin screw melt granulation process. For the purpose of the study, complete cone sigma line was used, followed by milling and tableting of the obtained granules. The authors use amorphous polymeric binders and paracetamol or hydrochloride as the model APIs. The use of thermoplastic binding with different properties was one of the key elements of the study, and some preliminary results excluded high viscosity grades. One of them was a finish oil, which is hydroxypropyl methyl cellulose. At the bottom table, we can see the different properties of the binders that used during the study, and includes the glass transition, the melting point of the binders, the solubility parameters, including the particle size distribution, and also the true density and the moisture content of these materials. The authors implemented the design of experiment with 31 different runs. As you can see on the right table, the polymer grade was investigated. The polymer concentration was also taken into account. The granulation temperature, the screw speed, and throughput of the process. The torque monitoring, the granule sphericity and moisture, also the tablet porosity and hardness were used to evaluate the whole process during the granulation and the tableting of the obtained granules. It was found that higher screw speed reduced the melting viscosity via shear thinning of the binder, and thus BA64, when it was used as a polymeric binder, resulted in successful granulation without barrier obstruction. For all formulations with high binder percentage and comparable thermal properties, the increase in the granulation temperature should be applied in order to optimize the process. The highest granular efficiencies were reached and obtained for the EPO and finish oil grades, and the milling process of these granules resulted in more fines. This can be seen on the top table on the right, where the particle size and of the milled and unmilled granules was recorded, and gives a full picture of the characteristics of the obtained granules. Minor differences were noticed in the tablet tensile strength between the melt granules and for both drugs. This can be seen in the middle graphs on the right, where we have the tablet ability of the two different drugs. The middle left graph is related to the tabletability properties of acetaminophen, and the middle right graph on the chlorothiazide tabletability properties. As you can see, for all different polymers, there was no significant difference on the tabletability, but when compared to the physical mixtures of the powder blends, we see a substantial difference. The porosity also of the obtained tablets was quite identical, again for both drug substances. We didn't observe any differences. There was a small difference on the paracetamol porosity, especially for EPO formulation, where they had a higher bonding strength at zero porosity. Also, during the study, it was found that high efficient breakup of the oversized granule fraction can take place, and the milling efficiency, as we mentioned before, was greater than 85%, with fines for most of the binders be around 6%. This is obvious on the graphs on the right part of the slide. Also, a Pca plot was implemented with PC1 differentiating the deformation mechanism, while PC2 in the y direction was used to capture the fragmentary behavior and correlated with the plastic deformation. So for the VA64 insolubles, it was found that both present plastic deformation and fragmentation, while EPO show a higher dielastic recovery after compression. I will introduce a twin screw granulation process that has been developed by Cubitech Extrusion and Delta Pharmaceutics, which is a spin-off company of the University of Greenwich, for the processing of Class 2 and Class 4 drug substances. This technology is based on co-processing of surfactants and solubilizers as granulating liquid with inorganic excipients, usually aluminum silicates and dicalcium phosphate hydrous, and of course the API. The technology presents several advantages and can be used for high drug loading of water-insoluble APIs, high throughput and continuous processing, and also a key element of technology is the downstream processing, which is minimized. Because of the nature of the technology, there is no need for fluidized bed drying, and the obtained granules present excellent flow and compaction properties, mainly due to the presence of the inorganic excipients. Further micronization can improve the granule properties, and an important feature of the technology is that the barrel temperatures can vary from 25 to 160 degrees. Eventually, the technology can be used to increase the dissolution rate of the insoluble APIs and thus the bioavailability. In this technology, the API should be highly soluble in surfactants or solubilizers, and the main advantage of the technology is that the solvents or the solubilizers are not volatile and can be used from 10 to 25%. These type of solvents or solubilizers are depicted on the bottom graph of the slide, where you can see a range of different materials. Usually these are grades of solubilizers or surfactants manufactured by Granulation, which is a French company. Each of them has a different HLB value. Of course, in order to identify the optimal and the best surfactant or solubilizer, we have to run some screening experiments to identify the material with the highest solubility of the API, and of course the miscibility of these surfactants. The co-processing of single solubilizers or surfactants or the mixtures can also take place. So in some cases we have to identify the ratio between the surfactants and solubilizers in order to optimize the process. Further evaluation can take place by using the lipid dialysis model, which gives us a very good idea about the solubilization capacity of these surfactants on the API, but also on the potential permeability of the drug substance. Overall, the twin screw granulation offers several advantages over traditional granulation approaches, and it's an ideal technology for continued manufacturing of pharmaceutical products. It meets regulatory guidelines as it's suitable for applying quality by design approaches and can be coupled with process analytical tools. It's a complex technology which requires systematic study to identify the critical processing parameters, critical quality attributes, and critical material attributes as well. And there are existing continuous lights made from different companies that can be effectively used for process scale-up. There's also a range of marketing products using twin screw granulation process that pave the way for future novel pharmaceutical formulations. I would like to thank our sponsors and the University of Greenwich, and also you for your attention and your time. I will be happy to answer any questions that you might have. Thank you very much.
Thank you, Dennis, for that informative presentation. Before we get to the Q&A, just a quick reminder to our audience that you can submit questions by typing them in the Q&A box, which is located on the right-hand side of your presentation window. So here's our first question. So how is the inline quality assurance achieved, such as drug content versus binder?
Yes, quality assurance can be achieved in different ways. So, for example, you can use inline equipment or offline equipment by testing the drug concentration of the solid state of the API or the crystallinity. In some cases, PAT tools have been used to test the polymorphic form, and also you can. There are other ways that down the process to control the humidity, or not control, but record the humidity in the fluidized bed, for example, or measure the particle size distribution of the granule, or even the morphology by using the right equipment. So there are different ways to assure the quality of the final product.
All right, thank you. Next question. What excipients have the largest likelihood of success in amorphization of poorly soluble crystalline drugs through TSG? What dictates this the most?
Yes.
TG's specific interactions?
Yes, I would say usually with extrusion granulation, twin screw granulation, it is not very easy to achieve complete amorphous state of the API. In that case, I would prefer to go with melt granulation or the technology. That was the reason, for example, we invented this technology, proprietary technology I present at the end of my presentation. So I would say, again, it's the miscibility between the polymer and the API, and of course, it's the operational temperature. You have to, if you use melt granulation, you have to go above the glass position of the polymer or the binder. We saw that in a study that was done by colleagues from the University of Ghent, where they tried different plasticizers. There were polymeric materials, and the applied temperatures quite high, above 140, 160 degrees, and those were above the glass position of the polymer. Again, I just want to repeat that it might be difficult to induce full amorphicity of the API by using granulation.
All right, thank you. Next question. Is there, excuse me, is there a relationship between the screwing speed and granulation temperature? I found the temperature was much higher than the settings when the screw speed was high. There's no control on that, and I fear about the degradation of the API.
Yes, this actually is a very good point, and indeed, by increasing the screw speed, we increase the granulation temperature due to the friction on the barrel walls, and this is a different situation. From our experience, I would say that we usually work around the screw configurations. For example, if you reduce the number of kneading elements or the kneading zones, or use different stagger angles, then you might have that effect on the increase of the temperature. And again, from my experience, it takes place usually in the kneading zones. So a remedy to this will be to play with the screw configuration.
All right, thank you. Next question. So this is about Raman as a PAT tool. Can you describe how the online probe was mounted to the extruder? And is the signal affected by the material load in the barrel?
Yeah, there are different ways to assemble the NIR or Raman probes. In many studies, the probes are placed on the top of the last kneading zone. We know for sure that it is affected by the temperature. So by changing the temperature or by increasing the temperature, the signal will be distorted. I'm not pretty sure if I expect a sort of small distortion of the signal, but not a great effect if we change the filling of the screws or the barrel. And there are other ways that in order to avoid the issues, there were some studies in literature where they actually place the NIR or Raman probes on the conveyor. So what they do, they discharge the material, the granules, from the extruder, and then they place on the conveyor, and this is where the probes are located to avoid any interference from the screw speed or the screw configuration. So I'd say these are the two different options to have a better recording or a better analysis of your granules.
All right, we have time for one more question. So can you describe the differences in formulations when working with paramesotol and VA64 in melt granulation versus hot melt extrusion?
Yes. In crystal granulation, there are a few differences between these two approaches. Actually, there's a completely different approach when we use crystal granulation and hot melt extrusion. So in crystal granulation, the amount of polymer we use is around 15 to 20%, and the API is still crystalline at the end of the process. In addition, the temperature is quite high, which is similar to that of hot melt extrusion, but only in the kneading zones. The common practice is to lower down the temperature in the last conveying zones, for example, around to 40 or even below 40 degrees to break any large agglomerates. So this is how we process the materials. We process coiled with paracetamol blends. In hot melt extrusion, the processing, the amount of the polymer is usually more than 50%, as the aim is to produce solid dispersions where the API should be amorphous at the end of the process. This is more evident in the water-insoluble APIs when we try to decrease the crystallinity of the API. In addition, the temperature profile that we apply in hot melt extrusion is quite high. We apply high temperatures in order to receive the exudates in the form of strands and to avoid blockage of the dye. So eventually, the temperature profile is quite different compared to twin screw granulation.
All right. Well, thank you very much, Dennis. Unfortunately, we are out of time. I'd like to thank the audience for attending and participating in this event. And I'd like to thank our sponsor, Thermo Fisher Scientific, for making this webcast possible. I'd like to invite everyone in the audience to participate in a brief survey, which you can find on the right side of your screen. And for our audience members, you'll also be receiving an email alerting you when this webcast will be available for replay. And I invite you to forward that announcement to your colleagues who may have missed today's live event. So thank you for joining us, and have a great day.